Hello everyone, this is Mrs. Sykes, and in today's video we're going to be talking about antigen variation. So this is part of the immunity topic and will be an A-level standard video. For those of you studying at GCSE, there will still be sections of this that are really helpful and will give you a wider understanding of the topic at large. So, this here is my pathogen, and on the outside of the surface of my pathogen I have antigens. And these antigens are usually made from protein. They can sometimes be made of glycoproteins as well, but the bog standard answer to be giving is that any antigen is made from protein. All living things have proteins on the outside of their cell membranes. However, it doesn't necessarily mean that they are fully turned into an antigen. An antigen, a foreign antigen, will cause an immune response. So to make this into an antigen, it needs to be able to trigger an immune response. So your white blood cells all need to be able to have a look at this, to be able to respond to this 3D shape, and once they have responded to that 3D shape, it will trigger them to go through the standard immune response. These antigens will be activating B cells. Now again, don't forget, we need to be very careful with the word activate when we're looking at immunity. B cell activation is a process. We would say that we stimulate cells in general, but we would only use the word activate when we're going through a very specific part of our cycle. So, antigens on the outside of our pathogen will help to cause the activation of a B cell. For this to happen, we need to have an antigen-antibody complex. An antibody-antigen complex is when we have those two things bound together. Now, the 3D structure of an antibody is really important, and its receptor site will be complementary to the shape of the antigen. So, in our simple Y diagram version, we've got our light and our heavy chains. Here is where we will find our receptor sites. And it will be here on this receptor site section where our antigen would bind. Not in here, as we would often show that at GCSE. It's at the ends, the fingertips of these arms is where we would find things working. So, if we are wanting to activate our B cells, the first thing that needs to happen is antigen recognition. Now, this can happen in two different ways. Firstly, the antigen can be found directly on the actual pathogen itself. Or you might find that an antigen-presenting cell, an APC, for example, this phagocyte here, has already found the pathogen and destroyed the pathogen and presented or shown the antigens that it has found on the outside surface membrane. And it will take these antigens and it will present them to the B cells and the T cells, the lymphocytes that we find in our lymph nodes. So this antigen presenting cell will travel to where all of the B cells and T cells are found. When we have the correct antigen, whether that's associated with the pathogen or whether it's on the APC, binding with the B cell, that will activate our B cell. So on the outside of our B cell, we have lots and lots of antibodies, which would all be identical to each other. When this B cell has been activated, it will then start going through the normal process that we would expect as part of an immune response. So we would have, firstly, mitosis is used to do clonal expansion. Immunity is a numbers game. If you can't produce enough B cells or T cells, you are going to be overridden by that pathogen and it may cause you to die. So the mitosis section of this is really important to get the number of B cells large enough to be able to make enough antibodies to get rid of the pathogen that we're dealing with. So our activated B cell, which has been activated by antigen recognition, goes through clonal expansion by mitosis. Some of those cells will be our memory cells, and they will stay in the body for a long time and will allow our body to recognise what is going on later on. But we will also produce plasma cells, and it is those plasma cells, short-lived, high energy expending cells, that actually give out our antibodies. And those antibodies will be an exact match for the ones on the original B cell and will be completely complementary to the antigen that we are trying to deal with. So we will be able to form new antigen-antibody complexes. All well and good. However, antigen variation is a thing. Diseases can change. Diseases can mutate. If these antigens here stay this shape forever, then the host, then perhaps the entire population, will remain immune to that disease, and that's the basis of herd immunity. However, if these antigens change in either size or shape just a little bit, it will mean that some of the population will lose some of its immunity, but some of the population will remain still immune. That little change will mean that some individuals will still be immune and some will not. This is called antigenic drift. Small changes, little changes to our herd immunity, little changes to the immunity of the population in general. However, sometimes the mutations that happen in terms of antigens can be fast changing, and those changes can be larger. So instead of just a little change in shape, you might have the equivalent of this happen, or maybe even this, or maybe even this. And as these changes occur, there is no way that those previous antibody complexes are going to work. This antigen is no longer complementary to all of those memory cells inside our body. Therefore, they are completely useless. So because we have had this antigenic shift, your body is no longer immune to this disease because it cannot recognise that it is one and the same. So an antigenic drift might mean that you're still immune to that disease, and it won't cause particularly big problems to the population at large. However, an antigenic shift is going to cause difficulties, not only for one person, but for the population. That will mean that no one is immune anymore. And if there is no immunity, that means that we are going to have many cases. So many people are going to be getting ill, many people are going to be contagious and pass those pathogens on to others. This could then cause a pandemic level event. I want to point out that this would be a pandemic event in the same way as the flu epidemics that we've had in 1918. This is not the same as what happened with COVID, because this is a pathogen we have met before that is now hidden from us because the antigens have changed shape. Things like COVID-19 was a brand new disease that our body and no one's body or immune system was familiar with. This antigenic variation is causing a loss of immunity. It is not because it is a novel disease. So, antigens are proteins. Those antigens are presented to B cells. When we have our correct antigen-antibody complex, it will cause the activation of the B cell. We'll go through mitosis and clonal expansion and create memory cells and our normal plasma cells, which produce antibodies. But if the antigens have changed, that immunity is lost. If it's a drift, it will be a little bit of immunity lost for a proportion of the population. If it is an antigenic shift, nearly every individual or every individual in the immunity will have lost their immunity. everybody will be susceptible to this, and that will cause spread on a large scale.